Good afternoon, everybody. My name's Azam. I'm the president and CEO of Tmunity, and I have the privilege of being the vice chair of the Alliance of Regenerative Medicine organization as well. And I'm really excited about the panel that we've assembled this afternoon. So we're going to be discussing the impact in large cap cell and gene therapy companies, particularly in relation to their innovation strategy. And over the past 3 years, we've certainly seen a wave of new players emerging in our exciting sector. And these are players that have obviously had a deliberate strategy of organic growth, also coupled with mergers and acquisitions. And hopefully during the time here in this exchange we're going to have with my guests, we're going to really get into sharing some of their experiences of the journeys they've been through, because I know they've all got a unique journey in terms of how they have got to within their organizations right now and how they have developed the portfolios and strategies now to hopefully benefit thousands, if not millions of patients around the world. So I'm delighted to have Bastiano, Krishnan, and Kathy and Jacob Sten, Peterson joining us today. So I'll just go around and let my panelists introduce themselves. So Kathy, why don't we start with you?
Okay, thanks, Oz. So I'm Kathy High. I'm president of AskBio Biopharmaceuticals, a wholly owned subsidiary of Bayer. And before that, I only started in this job a couple of months ago. I was a co-founder and head of R&D, president and head of R&D at Spark Therapeutics, which was acquired by Roche in December of 2019.
Thanks so much, Kathy. Jacob Sten Peterson.
Yes, hello. I'm Jacob Peterson, and I'm heading up a newly formed stem cell R&D unit at Novo Nordisk that I'm really excited to speak about a little bit later. Before that, I was responsible for Novo Nordisk diabetes and obesity research for almost a couple of decades.
Thanks, Jacob. Krishnan.
Hi, thanks, Oz. First of all, it's a pleasure to be here with such a distinguished group of individuals, so I'm really excited to be here. I represent Bristol-Myers Squibb. My role is I'm the cell therapy franchise lead, really representing the cross-functional enterprise view for cell therapy. Prior to Bristol-Myers Squibb, I came in through the Celgene Juno acquisition into BMS. So I have a little bit of legacy history in that I was in business development alliance where we were doing a lot of alliances and business development activities in cell therapies.
Thanks, Krishnan. And Bastiano.
And it's also a pleasure to be with you guys. I lead the cell and gene therapy division at Vertex. I came, I think, with a couple of us through an acquisition from the Sanna Therapeutics acquisition where I was CEO there. So Vertex Cell and Gene is an umbrella that has stem cells and gene therapies, as well as a couple of other things that we are doing in research.
Thanks so much, Bastiano. So guys, I think what would be great is if we kicked off just going around a little bit and really just getting the audience grounded on the strategic areas of innovation investments within your respective companies. And it would help also if you could explain why those particular areas are pursued, particular disease areas, your platforms, your product areas at high level. So Jacob, would you mind starting with a little bit of background about Novo Nordisk and the creation of your business there? And the real key areas of focus. You're on mute, Jacob.
Okay, sorry for this. So it will be a big pleasure. So Novo Nordisk have decades of experience within mammalian cell cultivation and manufacturing, and also within stem cells, in particular within type 1 diabetes. And based on this, we approximately, I think 3 years ago, decided that we will explore these capabilities much more broadly within pluripotent stem cells. And we formed an independent stem cell unit within Novo Nordisk that's operating a little bit like a biotech company within Novo Nordisk. We have now built expertise within the full R&D value chain, including a stem cell manufacturing site outside Fremont in San Francisco. We have added a device unit, which we think are important. and we are in the progress of building commercial capabilities. We have approximately 2 handful of projects. Several are on the way to the clinic within the next few years, and we are broadly interested in cell therapies based on pluripotent stem cells, and to some degree agnostic to the therapy area. Of course, we'd like to focus on areas that are close to what we are doing already in the cardiometabolic space, but we are also having therapy areas outside the traditional Novo Nordisk therapy areas. The only thing we have deselected, and that's for competitive reasons, because we don't have a track record and we have started late, that is cancer, at least for now. And one thing I think that's important is because we are so broadly interested in many therapy areas, most of the projects that we actually do, we do in collaboration with academia or biotech, so we in this way can build capabilities faster and certainly a lot better than we were able to do if we just had organic growth.
Thanks, Jacob. So Krishnan, it's been a busy time for you guys in BMS. I mean, 2 CAR-T approvals in close succession. Congratulations on that, and all coming in through various iterations and journeys into BMS, right? Celgene and also partnerships with Bluebird. But it'd be great if you could give us a snapshot of, you know, the main areas of focus and how you guys are thinking about platforms and specific areas of disease focus at BMS.
Yeah. Thanks, Oz. Yeah, I'd say, you know, how we got into cell therapy is a little bit driven by our legacy. You know, Celgene had a strong history in hematology. So as you can imagine, as leaders in hematology, that was a real key area of focus for us. And within that, we had a partnership with Juno, which helped us with CD19. And then given our interest in multiple myeloma, given our experience with Revlimid and Pomalyst, we were very interested in BCMA as an important target for myeloma, which led to our partnership with Bluebird Bio. And so as part of the acquisition, we came into Bristol-Myers Squibb. So, you know, our 2 key areas of strengths, I'd say, is hematology now and solid tumors, given our experience in IO with Opdivo at Bristol-Myers Squibb. So our real areas of therapeutic focus is really heme malignancies and solid tumors. And, you know, with the 2 product approvals this year, over the last couple of months, it's been a little hectic, to say the least, but getting Breyanzi approved and now getting that to patients is really exciting. And Abecma, you know, just last week in multiple myeloma is really an incredible feat. Our first BCMA CAR-T in multiple myeloma. So that's really from a disease area focus, really focused on heme malignancies. You know, we're hopeful about the future in solid tumors. Obviously, you know, we're not there yet, but an area of focus for sure. In terms of platforms, you know, we're really focused right now on both autologous and allogeneic. Having said that, I think our view right now is, you know, this is an area that's going to evolve over time. So as we think about the next 5-10 years, we really don't see this as, you know, one platform that's going to be the key platform. We think there's going to be an opportunity in the marketplace for both autologous therapies and allogeneic, but we need to do better with autologous products. So what are the next generation tools and technologies we can really build on to make the product faster, cheaper, more effective, is totally an area of focus, right? How do we automate things? How do we improve supply chain for autologous products? How do we make the products better? Because we're still not at a point where we're getting 100% complete remission rates. So how do we think about dual targeting CARs? So really focusing on making autologous products better, but also leveraging next generation with allogeneic investments.
Thanks, Krishnan. So Kathy, we were neighbors for a good couple of years in Philly, and now you're at AskBio. So I know that journey you took with Spark, but tell us a little bit about AskBio and your focus there now as part of the wider Bayer organization.
So, I can echo some of the comments actually that Krishnan made, but AskBio is also an AAV gene therapy company. And it was founded by Jude Samolsky, who's really been a pioneer in the molecular virology and vectorology of AAV. And I was attracted to the company because he's been able to translate a lot of his insights about the vectorology into very extensive manufacturing improvements, including a lot of experience with production in a suspension culture cell line, new experience with the use of synthetic DNA, called doggy bone DNA, as the raw material for making AAV. So instead of plasmid DNA, where you have to worry about contamination with bacterial DNA, bacterial proteins and so forth, just synthetic DNA. So much cleaner, faster timelines, faster cycles for manufacturing. So, some very attractive innovations in vector manufacturing. And then in terms of the pipeline and the programs, so it's a clinical stage company. There are 3 programs currently in the clinic. And what particularly attracted me is that not only do they have robust programs in single gene disorders, and I still feel that that's the bedrock of a gene therapy company, but also expanding out to gene-based strategies for complex acquired disorders, like, for example, heart failure. So a wide range of programs that I've found very attractive. And some of those overlap well with interests of Bayer, and some of them I think will be new areas of interest.
Excellent, thanks, Kathy. And Bastiano, great to see you again. It's been a while since we hang out together at Novartis, but you've been on an amazing journey these past few years and now landed at Vertex after the acquisition of Sanna. So tell us, what's the dream there in Vertex with cell and gene therapies? What are you guys thinking?
Absolutely. So I think I'm maybe a bit different than others and most other companies that I've also worked with in the past. At Vertex, we are somewhat platform or technology or approach, I would say, agnostic. We are a disease-focused company. So we focus on the disease first, and we commit to a particular disease, and then we look for the best tool to actually tackle that problem. Sometimes with redundant programs. We might have a gene therapy, a cell therapy, and a small molecule on the same disease. And we think about the diseases as diseases that need to have a strong human genetics fundamental, so that it kind of de-risks the approach. And we focus on diseases that are in general what you would call specialty diseases. So diseases for which there's not much of a need of a large commercial infrastructure. That allows us to pretty much channel about 70% of our budget into our R&D, which is quite high considering the size of the company. CF is one of them. So if you think about what I do for cell and gene, I have the most advanced programs are in type 1 diabetes, for which we use cells. Then we have hemoglobinopathies, sickle cell and beta tau, for which we use a combination of cell, HSCs, and gene editing. And then we have Duchenne, for which we use a viral vector. We also have a few things in research for which, again, we are platform agnostic. We do not commit to platforms. We think technology is a mean, is not an end.
That's great, Bastiano. Thanks. So, all coming from various journeys from big to small. In the case of Jacob, I think Novo Nordisk, you've clearly been there some time, but other colleagues having evolved into their respective organizations that's there now. Now, curious to ask you guys about your sort of focus when you think about geographies, because, in the small company world, right, especially your parochially, you have to think very focused. I mean, for example, in a company like mine, we're very right now focused on the US and getting a beachhead because of the base of the company and the programs are running. But curious to know from the lessons that you've learned in your past lives and now the organizations you're working in, how do you really think about your global focus? So, for example, in terms of feasibility of, say, from US being in Europe or Europe being in US, how are you thinking about access, trial execution? What are the kind of conversations you have internally as you sort of produce your algorithm of really defining focus? Because we all need focus, right? There are only so many resources we can use, and it's a competitive space. Cell and gene therapies for sure, and we all want to play to win for our patients. So, Krishnan, tell us how you're thinking about it. And you're in a huge organization now, but you clearly came from a mid-sized organization, right? Like Celgene and Evolve. So, love to hear your thoughts on how you're thinking about global positioning of your platforms and products as you develop them.
Yeah, it's a great question, Oz. I mean, I think we're probably still figuring things out, to be honest. As a global company, we're thinking about how do you efficiently bring these medicines globally. So by far, the immediate focus tends to be US, Europe, Japan. But, in other areas within BMS, generally the aim is to do simultaneous regulatory submissions. And, given the complexity of the cell and gene therapy space, it hasn't yet been feasible to do that. So I'd say as we think about, US, Europe, and other parts of the world, it's, supply, especially with autologous products. Do we have the infrastructure in place to ensure we can deliver these products appropriately in a rapid way, particularly in the disease areas we're focused on? So I think there's a lot of conversation about supply, capacity, how is that going to intersect, and particularly in an area where we're competing for slots, how do we ensure what we manage that process is really critical. And then I think the other important piece is, reimbursement and access. One thing is to get these medicines approved, but how do you actually get them accessible to patients? And so we're thinking a lot more earlier in the development programs about evidence generation. How do you show value to payers? I think these are things that are becoming even more critical, especially given the cost of cell and gene therapies. So I'd say, those are some of the key discussions we're going through now is around supply. How do we manage reimbursement, access challenges? And then, of course, global clinical trials is another big component of that.
Thanks, Krishnan. So Jacob, you touched a little bit in your opening, right, in terms of Novo Nordisk and how you've been thinking about globalization and footprint, and now with presence on the West Coast and the States as well from a R&D based perspective. It'd be great to get some insights for you. How are you thinking about your footprint, and how do you think about prioritizing and focus for the cutting edge areas you want to work in the stem cell space?
Yeah, I think as we just heard, Novo Nordisk is also a global company. So we will eventually execute our trials and commercialize globally. So that means that we, as soon as we can, will start global trials, and that's in the US, in EU, and Japan. And I think Japan, I would like to speak a little bit about that because there's a Sakigake legislation in Japan that will allow you basically to get conditional approval following a small, 10-20 patient clinical trial. So that's of course of interest, of course, provided that you can demonstrate safety and you're very sure of that. So that's why we actually have a strong focus also on Japan besides the US and EU. But the first phase 1/2 trial, since the regulatory environment in the US and Europe and Japan are so different, we kind of, I wouldn't say shop around, but we have interactions early on before we start with all 3 territories in order to figure out where is it the best possible condition to do a trial that would then lead to global clinical trials. So that we have a strong focus on, and that may sometimes, depending on indications, be in the US, sometimes in Japan, and other times in Europe. But as we just heard, we are learning. We are on a steep learning curve together with the regulatory authorities, I guess. And then also, as we heard, we have a strong focus on evidence generation. We believe that is important because as we develop these therapies, some of them will be very expensive, and we of course need to be sure that the patients will pay for the therapies that are actually working. So that we also have a strong focus on. But a little bit shopping around, trying to find where does it make most sense, but have a strong focus on Japan, because there are some possibilities that are simply not possible in the US and in Europe, and I don't think it probably will be possible. So that's our global strategy.
Thanks, Jacob. So Bastiano, sickle, thal, Duchenne, type 1 diabetes. So how do you think about all of this when you think about to prosecute, disease-focused product-specific strategies out of Vertex? Well, what's the algorithm? What are you guys thinking?
Yeah, so as you probably can see, I have the perverse pleasure of having all sorts of different approaches, because I have autologous cell therapy with CRISPR that has the vein-to-vein complications that we all know. I mean, as you know, us from our CAR-T days. I have an allo product for stem cells for the cure of type 1 diabetes. And I have an AAV9 type of approach, which has, of course, a different type of supply chain and manufacturing needs. Also, the diseases are somewhat geographically different. Beta thal is predominant in certain areas of the world, say the Mediterranean area in Europe, as well as certain areas in the rest of the in the world. Sickle cell tends to be, of course, a disease that is sub-Saharan Africa of origin. So in all those countries, like in the US, in England, in France. And of course, we're gonna take our medicines wherever the patients are. So there would be, we are a global company. We have trials open now for sickle cell in the US, for example, and in Europe, beta thal the same. And type 1 diabetes will take our medicines wherever the patients need them. So we are really not looking at this with a lens of where it's convenient to develop. It's more like, where are the patients, and we'll try to do our best to take those medicines to them, the same way we've done with CF, which is a disease that has pockets and everywhere in the world, and we do commercialize everywhere in the world.
Thanks, Bastiano. So Kathy, different times, different organization now, but still you've got the strong passion for science and patients, knowing you for all these years and how you've focused at SPARC, but now focusing in AskBio. How are you thinking now with the resources and infrastructure of a larger organization, but still very independent as a biotech and cutting edge biotech within Bio? How do you think about your global footprint and planning for the future?
Well, I have to say, Oz, that it's still very early days in this new organization for me. And I do remember that the chief medical officer at SPARC, who had come from Allergan, Dr. Kathleen Reep. So she had been a senior vice president at Allergan and had been involved in from the other side in many mergers and acquisitions, acquiring smaller companies. And I remember that she told me once, it takes about a year of an integration for you to really understand how things are going to play out. So with 12 weeks of experience, I'm not sure I can really comment.
Well, I can see Krishnan smiling, right? So maybe that's a man you should speak to, because I think he's got some experiences, right? From cell gene to DMS.
We can share some notes, Kathy.
Okay, great.
But early days, but early days. So, but exciting times as well, right? Because you've got a blank sheet of paper in some ways, right?
Yes, that's right.
Be able to figure this out and look at the organization that you need to adapt to. And so, another key area that I think we touched upon a little bit already is gene editing. So I remember back in, I think it was 2011, the years have gone by so quickly. 2011, 2012, actually, I was still at Novartis. So at that time, Bastiano may remember Intellia. Novartis was part of the investment in Intellia at the time. And I remember those deep discussions at CRISPR Therapeutics, Intellia, Editas. And that was the world, right? Now look at where we are, right? I mean, literally, there is not a week goes by where I'm not trying to educate myself on the latest base editing, the latest CRISPR-Cas enzyme system, the latest next best thing. And so the landscape has just become so rich. And I'm just going to make an assumption that we're all in organizations where gene editing is going to matter in some shape or form. So the question I'll ask, and Kathy, I'm going to start with you this time, is how do you really think about gaining access to the enabling technologies you need? Are you thinking of partnerships, M&As? I mean, because it's such a critical time for us all now in this space, but I'd love to get some perspectives from you guys in terms of how you're thinking with the rich ecosystem that we have now. How do you really think through enabling technologies like gene editing?
Well, I'm in a little bit of a difficult position here, Oz. I'm on the board of directors of CRISPR Therapeutics, and I'm therefore firewalled from anything being done at AskBio or Bayer on CRISPR. So you may have to edit out.
That's okay, Kathy. That's okay.
But I'll speak more generally. Please. About other technologies, perhaps not gene editing.
Absolutely.
But, I do think that, so I'll say a couple things. Number one, our company that's focused on AAV gene therapy has more in front of it than an organization 10 times its size could get done in the next 10 years. So when I think about these things, I do not feel absolutely compelled to follow each new platform.
Right.
Having said that, I will say that I think that there are clearly some indications where novel technologies like gene editing or other therapies that we can use in an adjuvant way with gene therapy are of great utility. And so I think of things that, for example, make it possible to dose people with pre-existing antibodies to AAV, technologies like that. So yes, it's a critical part of what we're doing in gene-based drug development to be aware of those other things. And I assume what you're referring to is that the universe of patents in gene editing is fairly closely held.
Correct. I mean, in the early days, there was the race to figure out the IP. We haven't still quite figured it out.
Yes, I would.
And now we have newer technologies, dare I say, potentially better. And so, the landscape just got crushed and crowded. And I remember, distinctly 4 or 5 years ago, thinking when we were doing a CRISPR-based program, do we take a license? Don't we take a license? And looking back in hindsight, I'm glad I didn't. But now, the decision I've got to make is probably the same as you guys, right? Well, what do I access and when, and at what price, clearly, and also supporting some of these great innovators, frankly, validating some of their early upstream technologies. So, I mean, it's an amazing time to be in this space, but it's also a real challenging time if you're a corporate development, business development colleague, trying to figure all of this out for the business. So, Bastiano, how's your head after all these years of looking at all of these interfaces? Because I know you have been looking. Bastiano, you're on mute.
We are somewhat agnostic again in terms of how we think about technology, because it's just a tool, and we think that, for example, we have a couple of programs in which the gene editing component of it is actually a critical component of the therapeutic approach, right? The program we have with CRISPR, of course, or the programs we have with that we're developing in Duchenne, where the actual gene editing is part of the therapeutic approach. And then there is a a variety of programs we have, either in early research or preclinical, where we use gene editing for as a tool to make modifications. It's not an integral part of the therapeutic approach, but is part of developing the medicine, right? So we take a look at all of those, and we use the one that is the most appropriate. As you rightfully pointed out, there is a range now of gene editing possibilities. There is single base, there is different type of enzymes, there is different types of ways of building your vector, your guides. We take a look at those that are the most appropriate for that program, and we take a look at those that are more appropriate, almost agnostically in terms of if they are internal, and we do have a fairly robust internal research program, or external, as you noted that we have a range of collaborations.
Right.
So I think is a very rich environment, and we think that the most appropriate tool will access it in any way that is necessary.
Yeah, the business drives the need, the enabling tool, basically. Yeah.
Correct. We don't get enamored of a particular technology to the point that we will use it all the time. Again, we have a range of tools. It's a toolbox, and we use the one that is the most appropriate.
Yeah. Krishnan, how are you thinking
Yeah,
about
I mean, I totally agree with what Bastiano just said. I mean, I think we have a range of tools in the toolbox, and we need to define which tool we need to be successful. And so, at BMS, we took an approach years ago to partner with Editas. And so that is a long-term relationship we have with Editas. And that was something we decided, and we amended that agreement to make that broader. But we continue to see kind of gene editing as a foundational technology for our cell therapy business. And so I think as we're thinking about where we are today versus where we were maybe, in 2015 is, we're going from maybe concerns around, freedom to operate before to how do you integrate the technology into the enterprise, right? How do we effectively and quickly integrate these gene editing technologies, from quickly validating the biological targets? How do you get quick, GMP raw materials? How do you optimize the manufacturing process consistently? How do you knock multiple genes in and multiple genes out? And how do you, and we're also seeing an evolution or the emerging requirements from regulators around these things. So I think we're really looking this as a tool in the toolbox, and we need to define, depending on for what platform we're focused on, which tool we need, but it's clearly going to be a foundational technology that we need to be successful.
Thanks, Krishnan. And Jacob, how are you thinking now in Novo Nordisk? Clearly with iPSC platforms, stem cell platforms, apologies. How are you thinking in terms of your strategy around editing or access to editing technologies?
Well, I think like the others spoke to, we use many different gene editing technologies depending on which phases. For research phases, it needs to be fast and quick, and the precision may not be that important. For the later development phases, when we're making the edits of our cells, we need much higher precision. So we also see it as different technologies and having a toolbox. But we do believe that stem cells and gene editing, you will need to have that in order to be competitive in the future. Probably not for the first generation products unless you're on CAR-T and other places, but if you are in more, in Parkinson's disease, chronic heart failure, type 1 diabetes, gene editing is probably going to come more in the second wave. And we basically see, 3 utilities within stem cell. The first one, which I guess everybody's working on, is to making a still cell line so you can avoid immunosuppression and the side effect that goes along with that. Because we believe by this you can attack much broader diseases and larger diseases where maybe the tolerance for side effects are significantly lower. For example, type 1 diabetes, at least in the general type 1 diabetes population, immune suppression would be unacceptable. So their stills could be important. The second thing we think will be competitive in terms of gene editing is to enhance the function of cells. So it could, for example, be cardiomyocytes where you add in growth factors that will either revascularize or regenerate heart muscle, besides contributing by itself to remuscularization. And the third, you can say, wave where we think gene editing will be important within stem cell is what we call forward programming or aiding differentiation. As most of you know, is cumbersome, is complicated. We spend a lot of money on expensive growth factors. And if you can do much of this by genetic editing, you can significantly lower the costs and hopefully also eventually the price patients will have to pay for our products. So those are kind of the three main things. And then in relation to IP, that is just a little bit a nightmare. We are trying as good as we can to get it settled out. We know many of the partners we work with, they more or less given up on this and say, you know, it will need to be settled later, and then they will take the appropriate licenses that they need. We are trying to a little bit more upfront to figure it out, but it's not easy. It's not easy.
Yeah, thanks for your honesty, Jacob. I feel the same when we think about IP. I think we all feel the same challenges, whether you're on the other side or the side of the innovator developing gene editing technologies or the acquirer,
exactly,
a licensor, we're all feeling the same. So, you know, a theme that came in, but I think it's an important one is, you know, the environment with regulators now. So, you know, I look back at, you know, what was a very unique moment in time, sort of that period between 2012 and 2017 and, you know, coming from the world I was in, which was the T-cell engineering space, you know, this, you know, real advancement in regulatory scientific thinking in terms of wanting to accelerate products. Clearly that also was in parallel happening with the gene therapies as well. Slightly different environment of late, you know, COVID had an impact, resources are being hit at the FDA. We know the challenges that colleagues at the FDA and global regulators, Europe as well, at the EMEA, and various other changes that have been going on. And then we see of late, maybe my perception, but a slightly shifting towards a more conservative stance, particularly in oncology. We've seen, you know, conditional approvals that have then been withdrawn because of lack of the data coming through, etc. But, you know, particularly in sort of PD-1 settings especially. But I'm curious to get perspectives from you as to how you're all thinking about individually in your own organizations around, does the regulatory environment really inform the way you're thinking about your disease and product strategies? So let's start with Krishnan first.
Well, thanks. Yeah, I mean, I think it's a really important question. I think we're all probably learning together kind of the evolving regulatory space. I think you're right. We are seeing that the regulatory environment is shifting. I think it's really this delicate balance of, you know, how do you get access to these therapies sooner while making sure you have sufficient data in the context of approvals. And so I think we're not really focused so much around, you know, which diseases we go into. I think it's more making sure we're really clear on the unmet medical need as we go into patient populations, because ultimately in the context of, you know, conditional or accelerated approval, clearly articulating the unmet medical need in the population is really important. So I think that's one area we're really focused on. And then frankly, really having the strength to make decisions if it's not really a game-changing innovation, you know, particularly now with the number of multiple CAR T products, many of them with the same targets, right? The number of CD19, BCMA for CAR T products that are now in the clinic. I think we're also really thinking long and hard about given, are you really going to demonstrate clear innovation here? Or is this just a small incremental benefit? Just given the cost of development of cell therapies today, I think we need to be able to say, if it's not a real innovation, to be able to kill programs early. So I think we're really refocusing our efforts to say, we need to be able to make sure that, clearly defined unmet medical need. And secondly, are you going to really demonstrate innovation in this space? And if not, having the courage to be able to say, we're not going to advance that therapy. So I think that's how we're thinking about this more broadly. And obviously this is important in the context of how quickly, these therapies can get to patients.
Thanks, Krishnan. So Kathy, your history and heritage in terms of the work you did at Spark, very precedent setting from a regulatory perspective, both when you think about the vector side, when you think about the patient population side, that, and unique moments in time. And how are you thinking about, do you think the regulatory, hurdles are going to be tougher now for the next decade moving forward now, as more focus on safety, gene editing? What's your thinking when you look at the regulatory landscape? Does it make you feel, any more uncomfortable than it was 5-6 years ago in the disease areas that you're looking at, or do you think that the regulators are going to be moving forward lock, stock, and barrel with us?
My own experience over a long period of time working with the regulators in cell and gene therapy on both sides of the Atlantic is that, to me, things continue to feel more straightforward than they were when we did the first trials of AAV in the late 1990s and early 2000s. And the accumulation of safety data, particularly around a single target tissue, has made working in those target tissues more straightforward. And I think that on the whole, my impression of the regulatory stance has been a nuanced one of benefit and risk, and that changes in the setting of diseases where there are already approved products. And so I think the advancement of cell and gene therapies for diseases in that setting has to meet a higher bar than in a setting where there is no other therapeutic alternative. But that on the whole, I've been encouraged by the fact that the regulators adopt a data-driven approach. So if you don't like their decision and you can come back and show them data why your stance is correct or better, my experience has been that they're willing to look at that.
Thanks, Kathy. And I think now the past couple of years, really the guidance is coming through now, getting more refined, more collaboration. I mean, certainly I felt in the cell and gene space, it's been a very unique environment to be in because of the high unmet need and the drive to want to get these products. So when you look at the metrics, I mean, even from the, some of the dialogues we've had within ARM, we look at, the regulatory framework and the number of products that are anticipated to be approved in the next few years, right? I mean, there is certainly momentum. But you're right. I think that the bar for data should be higher and is getting higher in terms of as these products get developed and get out to wider markets, then it behooves us as innovators, right, to think about that as well. Bastiano, sorry,
I'm
go
really
ahead.
glad you brought up the guidances, though, because I think those have made a tremendous difference, as well as the elimination of the duplicative oversight method that we had in the United States.
Yeah, for sure. Sebastiano, again, because of the complexity of the not so much the complexity, but the diversity of the different diseases that you're thinking about. Regulatory, is that something that gets you, gives you anxiety from a sort of portfolio and performance standpoint, or are you going with the flow and feeling that you're getting what you need from global regulators in a company like Vertex? Bastiano, you're on mute.
I don't think that anybody that does what we do is without anxiety when it comes to regulations. I think it's
Maybe I'm just transferring my anxiety onto you.
It's just, I think it just comes with the territory. It's just like, yeah, part of what we do. I think that what we're seeing is also a function of how much activity there has been in the space. I mean,
Yeah.
Regulators themselves now have much more experience because they see more programs. And they have the luxury of seeing every single program, as opposed to us, they only pretty much see our programs. So I think a lot of the guidance, a lot of the conversations that have been occurring is because there's much more understanding of what cell and gene therapy really look like, what has worked, what has not worked, both in terms of safety, in terms of analytics. So I think the regulators are just more experienced than they were a few years ago, even just a few years ago, without going back to the pioneering era. So that is definitely one aspect. Another aspect is something that I think both Krishnan and Katherine have touched upon, which is the concept of innovation and unmet need. I think that I haven't seen, at least personally, I haven't seen any sort of like roadblocks or unreasonable type of demands where the innovation is there and the unmet need is severe. I think that it might be a little bit more challenging for the third, the fourth, the fifth, program that is trying to do the same. The third aspect that maybe we haven't also touched upon is we all know, like, cell and gene therapy a few years ago, it was the realm of mostly oncology or ultra rare, vanishing small populations, usually of pediatric diseases with really, really awful options. Now is expanding, and is expanding to larger populations and is expanding to non-malignant populations. For example, at Vertex, it's very well known, we do not do oncology. And one of the programs I have is trying to get into a disease, type 1 diabetes, that is maybe generally considered not as lethal or as severe when it comes to the management as, for example, pediatric ALL, that, that's where we started, you and I started in CAR-Ts years ago. Now, we do believe that even for those patients, the unmet need is huge. That's why we are doing it. But in terms of your safety efficacy ratios might be a little bit different than, for example, an acute pediatric leukemia.
Thanks. And Jacob, how are you thinking from the world of stem cells and Novo Nordisk? Are you seeing you're getting what you need to progress your programs and advance with regulators?
Yeah, I think I will agree very much with a lot of what Bastiano just said. Type 1 diabetes, for example, there is, we have really, really good treatments that are relatively safe. So there's, and we're talking about young people, sometimes kids. There are really no rooms for any side effects whatsoever. So I think as the regulatory agencies gain more experience, more programs are coming through, there will be a steep increase in the demand, not only in terms of safety and tolerance for safety, as we also as industry learn, but also in terms of reproducibility and quality of the products that we are trying to develop. And consequently, I think at least for cell therapy, at least for some indications, there will be a move from autologous or donor-derived, you can say, umbilical cord, bone marrow cell therapy to more allogeneic pluripotent stem cells, because this will allow for better reproducibility, higher quality, and more importantly, also allow us to address diseases that are large, because we can do large-scale manufacturing serious chronic diseases like cardiovascular or Alzheimer's or other diseases where millions of patients are suffering, where we need large-scale capabilities of high reproducible quality product. So I think that's the way the field probably will move in the future.
So I've had a lot of fun hanging around with you guys, but I've just realized we've got 5 minutes left. So I'm going to give you each a crystal ball, and I'd like you to think about the future in the next 5 years and give us a prediction for the cell and gene space. How about that? So I'll kick it off. I think that in the next 5 years, we will see for the first time possibilities of cures with solid tumors, deep and meaningful sustained responses with T-cell based platforms. That's a prediction I'm making. Krishnan, what's your prediction with the crystal ball?
Yeah, I think we're going to see more waves of innovation, expanding patient pools. At least in the short term, I see, going into earlier lines of diseases, clear demonstration of efficacy there. But then longer term, I do see us moving with, single therapies, both autologous, probably to start with, in solid tumors with TCRs. We're already starting to see some early clinical data now. So I think I see us moving from heme to solid tumors in the oncology side. Yeah. I do see that as a real opportunity. And then frankly, I think gene therapy is going to expand even more opportunity for patients to have real cures.
You've got a great crystal ball there, Krishnan. Kathy, give us a prediction. Next 5 years, what do you see? You're on mute.
So I think that we will continue to see approval of products for single gene disorders. I think I'm very excited about the fact that I think we are going to see some diseases that have really not had good therapeutic options at all.
Yeah.
Are going to be diseases that people are going to be lucky enough to have treated and leave behind. So I think we'll continue to see approvals for single gene disorders. I remember that Scott Gottlieb, I think it was, when he was FDA commissioner, predicted that by about this time we were going to be seeing 20 cell and gene therapy approvals every year, or 40 or something like that. I'm not going to be that bold, but I do hope that we've reached the inflection point that monoclonal antibodies hit in about 1997, where then there was a really dramatic uptick in the number of approved therapies. And my other prediction is that I believe that within the next 5 years, we will begin to see approvals for complex acquired disorders like age-related macular degeneration, like heart failure, like Parkinson's disease, with cell and gene therapies that we have not had before.
Wouldn't it be great to see a breakthrough in CNS, right? Alzheimer's, big conditions like that, huge unmet need. Jacob, give us your crystal ball prediction.
Yeah, I think that as we go down the line, it's going to be more and more difficult to predict a different future. So I'm very much in line. I think that one of the big shifts that we spoke a little bit about is going to be not away from cancer, but we are going to add on top of cancer, where these cell therapies is very much focused. And we're going to see curative approaches demonstrating proof of concept in the clinic within Parkinson's, other CNS diseases, epilepsy, maybe type 1 diabetes, like we just spoke about, chronic heart failure, dry age, macular degeneration, and many other serious chronic large diseases. I hope we are going to see proof of concept in those.
Fingers crossed.
And then I will say that the latest prediction I read from the FDA was in 25, 10 to 20 therapies are going to be approved each year. And I don't hope that's going to move to 30, but let's hope for 25.
Hope so, right? Yeah. Bastiano, one prediction.
So as somebody more famous than me said, it's very difficult to make predictions, especially about the future. But what I hope and is what motivates me and my organization to do what we do, is that in 5 years we'll have a cure for type 1 diabetes, hemoglobinopathies, and Duchenne based on cell and gene, which will be an amazing
Amazing.
for those patients.
It's in reach. It's in reach, guys. We're in the golden era of medicine for sure. And I agree with Kathy that the inflection point we're at that inflection point now. So guys, Kathy, Bastiano, Jacob, Krishnan, thanks for giving your time today for this panel. Really enjoyed reconnecting with some of you, getting to know some of you, and thanks for the time.
Likewise.
Best of luck in your organizations and for the thousands of millions of patients you want to serve and get products to.
So much.
Thanks a lot.